Hello, everyone, and welcome to today's webinar, which is entitled Influenza mRNA Vaccines, Mechanisms and Methodologies. This is the first in a series of 4 webinars being brought to you by Jen and the CRISPR Journal that will cover some of the latest advances in RNA research and technology. This webinar series is being made possible through proud sponsorship from TriLink Biotechnologies. I'll be your host for today's event. I'm Jeff Pogaliskas, technical editor for Genetic Engineering and Biotechnology News, which has been at the vanguard of the life sciences industry for more than 40 years now, bringing you the latest information on the newest tools and technologies that can aid your research endeavors. Now, after more than a year of hearing about mRNA vaccines regularly in the news and in the scientific literature, and then, more recently, being able to finally witness their power and versatility, the scientific community, as well as the general public, are much more convinced about expanding their usage beyond just COVID vaccines. Now, prior to the coronavirus pandemic, mRNA vaccine research was well underway for use in combating other human diseases like cancer, HIV, and influenza. A virus our guest for today's event knows quite a bit about, and he's going to tell us much more about his laboratory's work on a universal influenza vaccine program that utilizes some cutting-edge methodologies. But before we dive into the data, let's learn a little bit more about today's guest speaker. I'm delighted today to welcome Dr. Norbert Pardi, who is an assistant professor of microbiology at the University of Pennsylvania, where his research interest is the development of mRNA-based therapeutics with a particular focus on new generation vaccines. Norbert has a strong background in biochemistry and genetics and has been at Penn since 2011, when he joined as a postdoc in the Weissman lab, where he continued working with his mentor, mRNA innovator, Dr. Catalin Carrico. During his tenure at Penn, Norbert explored the development of a novel therapeutic delivery platform using nucleoside-modified mRNA in lipid nanoparticles, or LNPs, and used it to generate highly effective vaccines targeting various pathogens. Now, this pioneering work is something he's going to talk a lot more about in just a few moments. But first, before we begin the webinar, I want to remind the audience that we'd love to hear from you in our Q&A session. Now, Q&A works a lot better if you send in questions during the event. You don't have to wait until the end of the webinar. You can send your question in at any time during the presentation. All you need to do is type your question into the Ask a Question box and then hit Submit. Norbert's going to be on hand to try to tackle as many of your questions as possible. Okay, now that all the housekeeping is complete, I'll hand things over to Norbert.
Thank you very much for the kind introduction, Jeff. Good morning, everybody in the US. Good afternoon, everybody in Europe. It's really a great pleasure to me to talk about my work today. My lab is primarily interested in the development of messenger RNA-based vaccines against infectious diseases, but we are also involved in the cancer vaccine field. We have several research programs that really focus on the better understanding of the mechanism of action of messenger RNA-based vaccines. Today, I'm going to tell you about our universal influenza vaccine program that also uses the nucleoside-modified messenger RNA vaccine platform. And in the second part of my talk, I'm going to share you some pretty recent data on the mechanism of action of mRNA-based vaccines. So we published the development of nucleoside-modified and purified messenger RNA lipid nanoparticle vaccine platform roughly 7 years ago. Let me very briefly tell you how we make these vaccines. So the messenger RNA is transcribed from a plasmid DNA template that has the antigen coding sequence. It has some special optimized 5' and 3' UTR elements and the poly A tail. We do co-transcriptional capping. So the cap1 structure, which is basically the clean cap, it's added to the 5' end of the messenger RNA during the in vitro transcription reaction. Once the RNA is made, it undergoes a stringent purification procedure. We use cellulose purification to get rid of double-stranded RNA and other types of contaminants that would otherwise be recognized as pathogen-associated molecular patterns, and they would induce unbounded immune responses. We replace uridines with 1-methyl pseudouridine in the RNA sequence to further increase protein production from the messenger RNA and decrease immune recognition of the messenger RNA. Once this purified and modified RNA is made, it gets formulated into lipid nanoparticles. This platform for infectious disease vaccine development, and this is actually the platform that is used in the very successful COVID-19 messenger RNA vaccines developed by Moderna and Pfizer-BioNTech. So let me tell you about our universal influenza vaccine program. As you all know, influenza virus infection causes significant morbidity and mortality every year. That can result in hundreds of thousands of deaths annually and tens of millions of cases. The issue is that currently used seasonal flu vaccines often provide very low level of protection. In some years, when the vaccine strains match with the circulating influenza virus strains, the effectiveness of these vaccines can be 50 or 60%, but in some years when the vaccine strains do not match well with the circulating influenza virus strains, the vaccines provide significantly lower level of protection. It can be as low as 10% or so. So there is an urgent need for the development of broadly protective influenza vaccines that protect from many different influenza virus strains, including potentially pandemic strains. So the messenger RNA lipid nanoparticle vaccine platform has many advantages over other vaccine platforms. One of the most critical advantages is the enormous flexibility of this vaccine platform that allows the co-formulation of several or sometimes many antigen encoding messenger RNAs into a single vaccine formulation. So we take advantage on this. In this vaccine program, we want to target several parts of the influenza virus, including the stalk or stem region of the major surface glycoprotein hemagglutinin of influenza virus. The reason why we really want to focus on the stalk or stem, because it's well established that it can induce broadly protective responses. We also want to include the neuraminidase, NA, the second surface glycoprotein of influenza virus, because it's also well documented that it's a fantastic antigen, and NA-specific antibodies can induce a high level of protection. Matrix protein 2, M2, it's also a good vaccine target. It's a critical part of the influenza virus. It encodes an ion channel. This is what we want to use in our program. So this is a beautiful ongoing collaboration between my lab at the University of Pennsylvania and several people at the Icahn School of Medicine at Mount Sinai in New York City. So our universal influenza vaccine program has basically 4 parts. The overall goal of the program is to target influenza A and influenza B viruses. The reason for that, because they are responsible for disease in humans. So influenza A viruses can be further divided into influenza A group 1 viruses and influenza A group 2 viruses. And we want to develop a vaccine against influenza B viruses. We are pretty much done with these programs, and right now we are working on the truly multivalent universal vaccine formulation. Today I'm going to tell you about our influenza A group 1 program. It's published, and our influenza B paper has recently been accepted for publication. So let's start with the influenza A group 1 studies. Like I told you, we included several influenza virus antigens in the combination vaccine. So we targeted the hemagglutinin glycoprotein of influenza A group 1 virus. And this was a stem targeting construct. That's why we call it mini-HA. And we used Y-type sequences from A Michigan 2015 H1N1 strain. The reason why we picked this because this is a recent vaccine strain. So we used the neuraminidase, the M2, and also the nucleoprotein from this virus. We decided to include the nucleoprotein in this vaccine because it's well established that this component is very conserved, so maybe it can induce broadly protective responses and NP-specific cytotoxic T-cell responses can contribute to vaccine protection. So the vaccination strategy was fairly simple. Animals received a single immunization with the 20 microgram mRNA LNP vaccine for each component. And 4 weeks later, we bled the animals and challenged them with influenza virus and monitored weight loss and survival. Before we started these studies, we wanted to make sure that each of the messenger RNA components in the combination vaccine works very well. So we performed an ELISA to measure antigen-specific IgG titers in mice that were immunized with the monovalent vaccines or the combination vaccine or a Firefly luciferase encoding RNA, which is basically an irrelevant formulation. And we were very pleased to see that we could induce antigen-specific immune responses with the monovalent vaccines and also with the combination vaccine, and we did not see a significant decrease in antibody responses induced by the different components of the combined vaccine. There was a little decrease in M2-specific antibodies in the combined vaccine compared to the monovalent vaccine, but it was really not a big issue. So we started the challenge studies with a heterologous strain. It's called IVR180. The reason why we picked this virus for challenge was that unfortunately the A Michigan 2015 is not lethal in mice, and we really wanted to use a virus that kills the animals. And you can see that the NA component of the vaccine, it's basically identical with the NA in the IVR180, but all other components are pretty different, particularly the HA component. So animals received a single immunization with the monovalent vaccines or the combination vaccine or the luciferase negative control vaccine. And 4 weeks later, they were challenged with 5 times LD50 IVR180 virus. This is what you can see on the left-hand side. And as you can appreciate, all the monovalent vaccines and the combination vaccine induced protective immune responses. Animals survived viral challenge, and it was we were very pleased to see that the neuraminidase vaccine and the combination vaccine induced protection without weight loss in these animals. In the next set of studies, we used the very high challenge dose, 500 times LD50. This is what you can see on the right-hand side. And we were very pleased to see that the NA vaccine and the combination vaccine induced protection even against this very high challenge dose. So next, we wanted to learn a little about the mechanism of protection. So we did a serum transfer study in mice. Animals received 2 immunizations 4 weeks apart, and 4 weeks after the second immunization, animals were bled. It was actually a terminal bleed, and serum was transferred to naive mice. And a couple of hours post serum transfer, these animals were challenged with the IVR180 virus. And again, we looked at body weight loss and survival over time. And as you can see, pretty much all vaccines except or all antibodies from different groups induce protection except the NP-specific antibodies. This is not very surprising. I told you before that the main reason why we have NP in the combined vaccine is that it can induce potent cytotoxic T-cell responses. So what we learned from this serum transfer study is that the protection induced by this vaccine is primarily mediated by antibodies. Next, we wanted to learn a little more about the mechanism of protection induced by these antibodies. So we performed microneutralization assays using serum from immunized mice, again, immunized either with the monovalent vaccines or the combination vaccine. And it turned out that only the neuraminidase component can induce neutralizing antibodies. This is, again, not very surprising because HA stem, it's reported that HA stem-specific antibodies and M2-specific antibodies often act through antibody-dependent cellular cytotoxicity activity. And this is exactly what we could measure. We performed ADCC assays using serum from these immunized mice, and it really turned out that M2-specific antibodies and HA stem-specific antibodies also have ADCC activity. We looked at the role, or we looked at the induction of T-cell responses by the NA and the NP vaccine. Animals received a single immunization intradermally, and 12 days later, animals were sacrificed, and we looked at antigen-specific NA or NP-specific T-cell responses, CD4 and CD8 T-cell responses in splenocytes. So the NA-specific responses are shown in the green bars, the NP-specific responses shown in the purple bars. And the take-home is that both vaccine induced antigen-specific CD4 and CD8 positive T-cell responses. And here in the G panel, you can appreciate that NP-specific responses were particularly, CD8 positive T-cell responses were particularly strong. We could measure high levels of interferon gamma producing NP-specific CD8 positive T-cells. So most probably it was a good decision to include the NP component in our combination vaccine. Next, we performed the dose de-escalation study and tried to determine the lowest protective dose induced by the neuraminidase vaccine or the combination vaccine. You can appreciate that even when we immunize mice with very low amounts, only 0.05 micrograms, so basically 50 nanogram NA vaccine or combination vaccine, we could still measure antigen-specific antibody responses. And in the lower panels, you can see that when we challenge these mice with the IVR180 virus, particularly in the case of the combination vaccine, you can see it on the right lower part of the slide, as little as 50 nanogram vaccine, combination vaccine after a single immunization could protect mice from losing weight. So this is a really, really potent vaccine formulation. Finally, immunized groups of mice with the monovalent vaccines, the negative control vaccine and the combination vaccine. And this was a single immunization, and we challenged these mice with the different influenza A viruses, and we monitored weight loss and survival in these various groups. And the take home, as you can see, all mice in the negative control group died after influenza virus infection, and the monovalent vaccines induced variable levels of protection against some monovalent vaccines were pretty protective against some viruses, but not against everything. And you can see the blue bars demonstrate that the combination vaccine induced very high level of protection. So basically there was a little weight loss after challenge, but it was also pretty minor. It provided broadly protective responses. So the take home from this part of my talk is that nucleoside-modified mRNA and LNP vaccination elicits strong antibody responses to multiple influenza antigens, and the combined vaccine protects mice from lethal challenge with diverse influenza A challenge strains. So let me tell you about our influenza B studies. The manuscript that describes these studies has very recently been accepted for publication. It's not out yet, but I'm going to tell you about the data. So our approach was very similar. We targeted HA, NA, and NP, and M2. We made monovalent vaccines, and we made a combined vaccine formulation. In this particular case, we used the full-length hemagglutinin because at the time of these studies, HA stem targeting influenza, an influenza B HA stem targeting construct was not available. So we used the full-length HAs, and actually the vaccine, the combined vaccine, has two HAs. The reason for that is that influenza B viruses have two distinct lineages, the Yamagata lineage and the Victoria lineage. So we picked one HA from each of these lineages. We used the HA from the B Phuket virus, it's a Yamagata virus, and HA from a B Colorado strain, it's a Victoria strain. So we used these two full-length HAs and the NA, MP, and M2 component from the B Colorado virus. We made monovalent vaccines, and we made the pentavalent combined vaccine, and we used the low dose 1 microgram from each antigen and the high dose 5 microgram from each antigen in these studies. And again, the luciferase was the irrelevant negative control vaccine in all of these experiments. Animals received intradermal immunizations, prime boost immunizations, so 1 or 2 doses, and we looked at antibody responses, and then we did several challenge studies using both Victoria and Yamagata lineages, and also the ancestral influenza B strain. So we were very pleased to see that, similar to the group 1 studies, a single immunization with the monovalent vaccines and also with the combination vaccine induced antigen-specific antibody responses as measured by ELISA. And again, it was great to see that in the pentavalent formulation there is no significant decrease in antibody responses against each of the vaccine components. And the second immunization significantly boosted immune responses as expected. Then we looked at the quality of antibodies induced by vaccinations, and we could measure neutralizing antibody response against the neuraminidase component. This is not surprising. And we could also measure neutralizing antibodies against the HA component of these vaccines. This was also expected because in these studies we use a full-length hemagglutinin. We expected to induce neutralizing antibodies against the head domain of hemagglutinin. So this is exactly what we see: neutralizing antibodies, anti-HA and anti-NA antibodies, both in the monovalent and in the combination vaccine, and these responses were boosted after administration of a second vaccine dose. We could also measure antibodies with antibody-dependent cellular cytotoxicity activity. We could measure it in the anti-HA and anti-NA antibodies after a single immunization. And interestingly, anti-M2 and anti-NP specific antibodies also had some ADCC activity after administration of a second vaccine dose. So basically, the take-home from these studies that immunization with the influenza B vaccine antigens can induce functional antibodies. Next, we performed a bunch of challenge studies. Again, groups of mice were immunized with the monovalent vaccines or the combination vaccine, and then challenged with the ancestral influenza B strain or various Victoria and Yamagata viruses. I know that this is a pretty busy slide. The take-home is that the monovalent vaccines induce variable levels of protection. So many times, even if the animals were protected, there was some weight loss. M2 and NP vaccines induce really variable levels of protection. Sometimes they were not protective at all against some viruses. But the most important finding is that the pentavalent vaccine, even in the low dose, so 1 microgram for each component, induced protective immune responses. So no death and many times minimal weight loss. So these vaccines are really, truly broadly protective. To demonstrate that the combined vaccine can induce sterilizing protection, we immunized groups of mice with the monovalent and the pentavalent vaccine, and then sacrificed one group at 3 days. And then 4 weeks post-immunization, we challenged these mice with the Victoria and the Yamagata strain. And then 3 days later, 3 days post-infection, groups of mice were sacrificed, and 6 days post-infection, another group of mice was sacrificed. And we looked at viral titers in the lungs of these animals by plaque assay. And the take home is that the combination vaccine, again, the monovalent vaccines induced variable levels of protection. Sometimes they could not protect from viral replication in the lungs. In some cases, they were more protective. The HA and the NA vaccines worked somewhat better than the NP or the M2 vaccines. But the most important finding that the pentavalent vaccines, and again, even the low dose, could induce sterilizing protection 3 days and 6 days post-infection against Victoria and Yamagata virus. So again, this was really, really great news to see this sterilizing immunity. Similarly to the group 1 studies, we performed a dose de-escalation study. We determined the lowest protective dose, and we only used the combination vaccine in this particular experiment. So animals received a single immunization with 5 microgram, 0.5 microgram, or 50 nanogram, and 5 nanogram combined vaccine, or the luciferase negative control. We looked at ELISA titers, and again, we could still measure titers after immunization with only 5 nanogram combination vaccine. And then we challenged these mice with the B. Malaysia strain and looked at morbidity and survival. And the take home is that as little as 50 nanogram combination vaccine could induce protective immune responses in these challenged animals. So it's really highly protective after administration of a single dose. We also performed a passive transfer study to see if antibodies can induce protection from viral infection. And in the top panels, you can see the animals received 2 immunizations. 4 weeks after the second immunization, we collected serum from these animals. On the left-hand side, you can see the ELISA titers measured in the donor serum. So every single vaccine induced antibodies. The M2 specific antibodies had a pretty low level. And then we transferred this serum from immunized mice to naive mice and challenged them a couple of hours post-serum transfer. Before challenge, they were bled, and we measured antibody titers in the recipient sera as well. And you can appreciate that obviously compared to the donor serum, there is a decrease in antibody titers. And again, 4-hour serum transfer animals were challenged with the Victoria virus, and we measured morbidity and mortality, and it turned out that NA and HA specific antibodies can induce protective immune responses against particularly the serum from the animals that received the combined vaccine was really highly protective. NP and M2 specific antibodies did not induce a very high level of protection from viral infection. So the take home from this experiment is that the protection induced by these influenza B mRNA vaccines is primarily antibody mediated. So the conclusions from this study is that most of the monovalent vaccines, influenza B vaccines, protected mice from infection after a single vaccination. Minimal or no weight loss was observed in mice immunized with the pentavalent vaccine. A very low dose, only 50 nanogram single immunization, can protect mice from influenza B infection, and the pentavalent vaccine formulation induced broadly protective responses after a single immunization. So I'd like to thank the contribution of many of my great collaborators. Jamili and Matthew significantly contributed to the group 1 studies. Hiromi and Csaba from my lab contributed to the influenza B studies. Raphael Nachbagauer and Alec Frein were absolutely key in the group 1 studies from Mount Sinai, and Megan McMahon really spearheaded the influenza B studies from Sinai. And of course, Acuitas Therapeutics, they are very long-term collaborators. They provided the lipid nanoparticle formulations. So let me change gears now and talk about pretty different topic. I would like to share some data on the mechanism of action of nucleoside-modified and purified messenger RNA lipid nanoparticle vaccines. As I told you, this vaccine has two basic components, the modified and purified RNA and the lipid nanoparticle. And it's very well established that the platform works very well, both in preclinical models and also in humans, as it turned out in the context of COVID-19 mRNA vaccines. But we know very little about the mechanism of action of these vaccines. So we really don't know what signaling pathways are involved in immune sensing, for example, of mRNA-LNP vaccines. And I do believe that this is very important. It's very important to learn more about it, because once we understand how these vaccines work, we should be able to come up with even more potent mRNA vaccines. So a number of years ago, we asked a very simple question: what provides the adjuvant activity for nucleoside-modified and purified mRNA-LNP vaccines? It's a very simple question, but actually we don't know we didn't know the answer. So all vaccines have some kind of an adjuvant activity. For example, in viral vaccines, either live or inactivated virus, it has an inherent adjuvant activity. The components of the virus can stimulate immune responses. In a subunit, protein subunit vaccine, we need to use adjuvants such as alum or MF59 to be able to induce strong immune responses. If you use an unmodified RNA vaccine, we do know that the RNA has inherent adjuvant activity, so it is recognized by various immune sensors. But the nucleoside-modified RNA was really designed not to induce strong inflammatory responses. It's an important question, and it was really important to figure out what is the adjuvant component in these vaccines. So to give a pretty quick answer, I put together a very simple experiment. So I used protein subunit vaccine, and the protein was a recombinant PR8 hemagglutinin protein. So this was basically an influenza vaccine, and BOBC mice received a single intramuscular immunization with 10 microgram protein, either protein without any adjuvant or protein mixed with empty lipid nanoparticles, or firefly luciferase encoding messenger RNA lipid nanoparticle construct. 4 weeks and 8 weeks after a single immunization, animals were bled, and hemagglutination inhibition titers were determined in these serum samples. And it turned out that, as expected, the HA protein without an adjuvant did not induce measurable HAI titers, but the MT-LNP adjuvanted or the luciferase mRNA-LNP adjuvanted vaccine induced pretty high HAI titers. And there was no difference in the HAI titers between the MT-LNP adjuvanted and the mRNA-LNP adjuvanted protein vaccine. So the take home from this experiment was that the lipid nanoparticles have adjuvant activity. So we repeated these studies a little later, and we added a comparator, an HA protein subunit vaccine adjuvanted with Adavox. It's a very similar adjuvant to MF59, which is basically a licensed FDA-approved adjuvants. So similarly to the previous study, animals received a single immunization with 10 microgram HA protein without an adjuvant, with the Adavox adjuvant, with the MT-LNP or the luciferase mRNA-LNP, and we followed the HAI titers, hemagglutination inhibition titers for 20 weeks. And as you can appreciate, the LNP adjuvanted vaccines or vaccine or the mRNA-LNP adjuvanted vaccine induced higher level of HAI titers compared to the Adavox adjuvanted protein subunit vaccine. So it's well established. We and others showed several years ago that nucleoside-modified mRNA-LNP vaccines can induce potent T follicular helper cell responses. What are T follicular helper cells? T follicular helper cells are activated CD4 positive T cells that help to form and maintain germinal centers within the B cell follicles. Germinal centers are the microanatomical sites of antibody affinity maturation. So TFH cells provide help for B cells. They provide signals, IL-4, IL-21, and they drive antibody affinity maturation. They also contribute to long-lived plasma cell formation and B-cell memory formation. So essentially, if you want to develop a vaccine that can induce strong antibody responses, you need to find a platform that can induce potent T follicular helper cell responses. And again, it's well established that nucleoside-modified mRNA-LNP vaccines can induce strong TFH cell responses, and in the subsequent studies, we used TFH cell responses as a readout. So we immunized mice with the hemagglutinin protein subunit vaccines, adjuvanted either with AdavoX or the MT-LNP, and we used HA mRNA-LNP vaccine as a positive control. Again, I told you that we did know that it could induce strong TFH cell responses. The question was if LNP adjuvanted protein subunit vaccines can induce also strong TFH cell responses. In all of these studies, animals received a single immunization with 10 microgram protein, and animals were sacrificed 10 days post-immunization, and TFH cell responses were measured in the draining lymph nodes of these animals. In the B panel, you can see the total number of TFH cells, and you can appreciate that LNP adjuvanted vaccines induced higher numbers of TFH cells compared to the AdavoX adjuvanted vaccine. And in the lower panel, in the D panel, you can see the hemagglutinin specific T follicular helper cell responses, and the difference is really striking between the LNP adjuvanted protein and mRNA compared to the AdavoX adjuvanted protein vaccine. So again, LNP adjuvanted vaccines can induce strong T follicular helper cell responses. We also looked at germinal center B cell responses, so GCB cells in these animals, again in the lymph nodes, and take home was very similar. LNP adjuvanted protein vaccines and mRNA vaccines can induce stronger GCB cell responses compared to the AdavoX adjuvanted protein vaccine. We immunized groups of mice with HA AdavoX and HA LNP protein subunit vaccine, and let them sit for a while for several months, and then and then we looked at plasma cell responses and memory B cell responses. So we really wanted to wait until we can measure these longer term responses. And it turned out that the LNP adjuvanted recombinant HA vaccine induced much stronger hemagglutinin specific plasma cell responses and memory B cell responses compared to the AdavoX adjuvanted protein subunit vaccine. So again, LNP adjuvanted vaccines can induce stronger T follicular helper cell, germinal center B cell, plasma cell, and memory B cell responses than the AdavoX adjuvanted HA protein vaccines. So we, in the next set of studies, we wanted to demonstrate that that LNPs can be used with other protein antigens as well. So not only with the influenza hemagglutinin. So we repeated these studies with another protein subunit vaccines, and this was the receptor binding domain of the spike glycoprotein of SARS-CoV-2. So the experimental setup was very similar. Mice received a single immunization, 10 microgram RBD protein with MT-LNP, 10 microgram RBD protein with AdavoX or 10 microgram RBD mRNA-LNP. And in the A panel, you can see that we measured antigen-specific IgG titers by ELISA, and you can appreciate that LNP adjuvanted vaccines shown with green and pink induced higher levels of RBD-specific IgG compared to the AdavoX adjuvanted protein vaccine shown with blue. We measured the neutralization ability of vaccine-induced antibodies, and again, AdavoX protein vaccines did not induce measurable levels of neutralizing antibodies, but the LNP adjuvanted RBD vaccine and the mRNA-LNP RBD vaccine induced pretty high levels of neutralization titer as measured by pseudovirus neutralization assay. Similarly to the HA studies, we found that LNP adjuvanted protein and mRNA vaccines induce higher level of T follicular helper cell responses, germinal center B cell responses, plasma cell responses, and memory B cell responses compared to the AdavoX adjuvanted protein subunit vaccine, the RBD vaccine. So the take-home message from this part is that that LNP can be used with not only with RNA but also with proteins to induce strong antibody responses and cellular immune responses, and the LNP component of the vaccine, mRNA vaccines, provides very strong adjuvant activity. So we went on and we wanted to determine which component of the LNP lipid nanoparticle is responsible for the adjuvant activity. The lipid nanoparticles that we use have 4 different components, and probably the most critical component is the so-called ionizable cationic lipid that is critical for the safety feature of lipid nanoparticles, and it plays a role in the uptake of mRNA LNP vaccines. So we asked our collaborator, Acuitas Therapeutics, that provide these lipid nanoparticles for us to generate a lipid nanoparticle, empty lipid nanoparticle with the ionizable lipid, empty lipid nanoparticle without the ionizable lipid. This is shown with brown. You can see that the size of these particles are roughly similar. The empty LNPs without the ionizable lipid are a little bigger. And so we repeated our immunization studies, and we immunized mice with the recombinant HA protein adjuvanted with the empty LNP with the ionizable or empty LNP without the ionizable lipid. And we measured hemagglutination inhibition titers, and we were very surprised to see that if we use an empty LNP without the ionizable lipid, basically it loses all the adjuvant activity. So we could not measure any HAI titers 4 weeks or 8 weeks post single immunization with these vaccines. So the ionizable lipid is critical for the adjuvant activity of LNPs. Next, we looked at the cytokine and chemokine profile induced by AdavoX adjuvant, the empty LNP adjuvant, the empty LNP without the ionizable lipid, and the luciferase mRNA LNP, and have used LPS as a positive control. Animals received a single immunization with a single injection with these compounds, and then we looked at several cytokines, or actually many cytokines and chemokines, 4 hours or 24 hours post-injection in the draining lymph nodes of these animals. The take-home message is that the empty LNP with the ionizable lipid and the mRNA LNP could induce, again, very potent production of several cytokines and chemokines, and we did not see it with the empty LNP without the ionizable lipid, and the AdavoX induced actually lower amount from these cytokines and chemokines. IL-6 was particularly interesting for us because we did measure high production of IL-6 by empty LNP and luciferase mRNA LNP, and IL-6 is a key regulator of TFH cell biology in mice. So we were particularly happy to see this, and we wanted to take a closer look at the role of IL-6 in mechanistic studies. So first we immunized the wild-type mice with the recombinant HA plus AdavoX and recombinant HA plus empty LNPs, and we looked at the kinetics of IL-6 production. So we looked in the draining lymph nodes of these animals. So we measured it at 4 hours, 8 hours, 12 hours, 24 hours, and 48 hours post-immunization or post-injection in groups of mice. And the take-home is that there is a very strong IL-6 wave after immunization with the LNP adjuvanted hemagglutinin vaccine. The peak is at 4 hours, and then there is a gradual decrease, and basically there is almost none 2 days after immunization. To further investigate the role of IL-6, we performed immunization experiments in IL-6 knockout mice, So animals were immunized with the HA-LNP or the HA-mRNA-LNP vaccine, and the TFH cell responses were measured 10 days after a single immunization, and it turned out that that TFH cell responses are really blunted in IL-6 knockout mice compared to the control wild type mice. So this is evidence that this provides some evidence that LNP-induced IL-6 is critical for the induction of strong T follicular helper cell responses by LNP adjuvanted vaccine formats, both RNA and protein. We obtained very similar data in IL-6 knockout mice when we looked at germinal-centered B cell responses. Again, germinal-centered B cell responses were blunted in IL-6 knockout mice after HA protein and LNP or HA-mRNA-LNP immunization. To provide some more evidence for the critical role of IL-6, we performed an experiment in wild-type biopsy mice. Animals received a treatment with an anti-IL-6 antibody or a isotype control antibody 24 hours before immunization with an HA-MT-LNP vaccine. Then they received this immunization with the HA-LNP vaccine, and we looked at the T follicular helper cell responses, and it's very clear that the TFH cell responses are blunted when we block IL-6 with the blocking antibody. So again, the take home is that LNP-induced IL-6 is very important for the induction of strong T follicular helper cell responses in mice. Next, we wanted to learn a little more about the sensing mechanisms. As you know, there are many RNA sensing receptors in different cellular compartments. And we can sense different forms of single-stranded RNA, we can sense double-stranded RNA. So we really wanted to learn a little more about it. And this is ongoing work. I'm going to show you just 2 or 3 slides about our initial studies, but we are still working on the better understanding of mRNA-LNP sensing by our immune system and how these vaccines can induce an immune response. So in the first set of study, we used the mAbs knockout mice. So mAbs is a common adapter for RIG-I, MDA5, and many other receptors that can sense RNA, different types of RNAs. And actually it turned out that when we immunized mAbs mice or control wild-type mice with HA protein plus MT-LNP or HA-mRNA-LNP vaccines, and look at T follicular helper cell and germinal-centered B cell responses, we could not see any difference between the control mice and the mAbs knockout mice. So the take-home is that mRNA-LNP vaccines are not sensed by receptors that act through mAbs. We looked at the role of another adapter, important adapter molecule, MyD88. It's a common adapter for toll-like receptors that can sense RNA. And the experimental setup was very similar. So animals received a single immunization with the HA-LNP or HA-mRNA-LNP. And we looked at T follicular helper cell, germinal-centered B cell responses 10 days after a single immunization. And it turned out that we really could not measure strong or significant differences when we used between the mAbs mice and the control mice when we used the HA plus MT-LNP, but we did see a blunted TFH cell response and germinal B cell response when we immunized mice with the HA-mRNA-LNP. So the take home is that most probably the mRNA component of mRNA-LNP vaccines is sensed by receptors, one receptor or more receptors that act through the MyD88 adapters. So these are ongoing studies, and I really hope to learn more about the sensing mechanisms in the near future. So to wrap up the second part of my talk, the LNP component of modified mRNA-LNP vaccines is a strong T follicular helper cell, B cell, and humoral immune response inducing adjuvant. This is, I believe, a very important discovery, and others also have very similar findings. 2 beautiful papers were published in Nature Immunology from Ira Mellman and Bolly Pulendran's lab, and with similar findings. We also found that LNPs can be combined with protein subunits to create potent novel vaccines. So it's a very good adjuvant that works not only with mRNA, but also with protein. The ionizable lipid component of LNPs is responsible for the adjuvant activity of LNPs. LNP-induced IL-6 is a major driver of TFH cell differentiation. And finally, LNPs are not sensed by either RIG-I or MDA5 or receptors that directly or indirectly rely on MyD88, but the RNA component or the degradative products of RNA are recognized by toll-like receptors and maybe other receptors that act through MyD88. So I'd like to thank all people who contributed to these studies, Matthew Pine and Hiromi Muramatsu from my lab, several people from Drew Weissman's lab, and I'd like to mention Michela Locci because her group, particularly Emily Bettini, was absolutely key in these studies. Emily and Michela performed all the TFH cell and GCB cell studies. They were really, really important, and again, I'd like to thank everybody else in this, everybody else for their contributions. So thank you very much for listening, and I'm very, very happy to take questions. Thank you.
Norbert, really, truly amazing work you're doing, and the data is really, really compelling. So I'm really looking forward to your future endeavors, as well as hopefully being able to take a universal influenza vaccine sometime very, very soon. To the audience, the Q&A is coming up in just a few moments. So if you have a question for Norbert about his presentation, which it looks like many of you do already, if anyone's sitting there thinking about asking a question, don't hesitate. All you need to do is type it into the Ask a Question box and hit submit. Okay, since we have a ton of questions already, I'm going to start the Q&A in just a moment. But if you still have a question, don't hesitate. Just type it in and hit submit. All right, bear with us for just a moment as we transition into the Q&A session.
All right, thanks everyone for joining us for the Q&A session. Norbert, again, great presentation. Really appreciate that. We have a bunch of questions here for you, so let's try to get through as many of them as we can. The first one we have, an audience member states, if we formulate a 10 to 12 mRNAs, or if we formulate 10 to 12 mRNAs in a single mRNA LNP vaccine, can we induce potent responses against each component in humans?
Hi everyone. It's a great question. What we found in animal studies that it's possible to induce very good immune responses against 10 to 12 mRNA encoded antigens. When we use them in a single vaccine, I think in humans, the major concern is the dose, because if you have 10 to 12 components, this means that most probably using the current formulations and thinking of the Pfizer or the Moderna vaccine, we cannot really use more than 5 to 10 micrograms per component. And I think the big question is if we can induce strong enough immune responses with, let's say, 5 to 10 microgram messenger RNA. So far, it seems that at least in those vaccines that we have tried so far, that different mRNAs do not interfere with each other. So this is not a concern, but again, the big question is the dose, because I doubt that we can use, again, based on the COVID-19 mRNA vaccines, I doubt that we can use more than 100 microgram mRNA in a single formulation. And again, if you in a COVID vaccine, it's only the spike mRNA, but if you want to use maybe 10 of them, then again the big question is really that the relatively low dose is sufficient to induce strong enough immune responses. You need to test it in human trials.
All right, great, thanks, Norbert. Next question we have, this pretty interesting one. An audience member says the current egg-based flu vaccine has about 40% protection. Do you think mRNA vaccines can reach 90% protection like COVID using the mRNA?
This is an excellent question, excellent point. At the moment, I don't think that this is a realistic expectation. Influenza virus is a pretty complicated vaccine target. There are several issues, but one of the most important issues is that most of us have pre-existing immunity against influenza, and it's pretty well documented that pre-existing influenza specific immunity can have very often a detrimental effect on vaccine-induced immune responses, and I think this is a main issue right now, and I'm really not sure if mRNA vaccines can really overcome this problem. I think we can definitely come up with at least as good mRNA vaccines as egg-based flu vaccines, maybe even better. And I think we still have like a big advantage, and this is the ease of production, and it's very easy to come up with a modified RNA vaccine. The production of these vaccines are much simpler. Again, if for those people, so for most of us who have pre-existing immunity against flu, I really don't think that 90% efficacy is at least with the current platforms that we use. I don't think that we can reach it. It would be very interesting to see how, for example, a universal mRNA-based influenza vaccine would work in very young children. Young children who have who do not have pre-existing antibodies. It would be really interesting to see if we can induce, let's say, using a multi-component universal influenza vaccine that I just talked about. It would be interesting to see how those vaccines would work again in this population without pre-existing antibodies.
All right, thanks, Norbert. Next question, can you compare SAM, circular RNA, and current traditional linear mRNA with respect to production and safety and efficacy?
It's a great point. I think in terms of production, these are mRNA. So all of these are synthetic platforms. So I think the COVID-19 pandemic really showed us that it's possible to produce these vaccines in very large quantities. I do not think that this is an issue with any of these platforms. Maybe one consideration that the self-amplifying RNA it contains not only the antigen of interest but also other components. So these RNAs are usually pretty long. So I can imagine that it may cause some issues with production. I'm not sure about that. In terms of the safety and efficacy, so the circular RNA is still a relatively new platform, so we do not have any human data on that. Based on animal studies, non-human primate studies, and mouse studies, it seems that it works pretty well, and people didn't see any major adverse events after using circular RNA. But again, this is only preclinical work. We have some data, some human data with the self-amplifying RNA, and it seems that this is a very promising platform, but most probably it needs to be optimized, further optimized for humans, because it works very well, particularly in mice. It also works in non-human primates, but based on some recent publications and at least in the context of COVID-19 mRNA vaccines, it turned out that they did not work that well in humans, and we need to understand why this was happening. And we have probably the vast majority of the data. It's the non-replicating RNA
both unmodified RNA and nucleoside-modified RNA. So for the nucleoside-modified RNA, I think the answer is pretty simple. The human trials, phase 3 trials, provided evidence, both the Pfizer and the Moderna trial, that nucleoside-modified RNA it's pretty safe and very effective, at least in the context of COVID-19 vaccines. The unmodified platform it was not as good as the nucleoside-modified, but I can imagine that it is possible that we can increase both the efficacy and also the safety profile of these of the unmodified RNA vaccine. Maybe that CureVac is a good example. They published their phase 3 data with the COVID-19 vaccine, and it was not as good as the nucleoside-modified RNA. But again, there is probably still some room for improvement. Just one more sentence for the nucleoside-modified RNA for the safety. I think it's safe, but many people experienced reactogenicity. So for the future generation of next generation of nucleoside-modified mRNA-LNP vaccines, it will be very important to decrease these adverse events after vaccine administration.
All right, thank you, Norbert. Next question is what antigens are taken in combination vaccine, and do you need a combination of mRNAs or one mRNA encoding all antigens?
Let me start with the second part of the second question. So we make individual messenger RNAs. If we talk about the flu vaccine, so we make the messenger RNA encoding the HA, NA, NP, M2 components, and then we combine these messenger RNAs before formulation to lipid nanoparticles. So we don't use a single messenger RNA encoding all of these vaccine components, but instead we make individual messenger RNAs, mix them together, and then formulate them into lipid nanoparticles.
All right, thank you for that. Next question we have, this is a very good question, interesting one as well too, and they wanted to know if you attempted intranasal immunization.
Yeah, that's a great point. So at the moment, the messenger RNA lipid nanoparticle vaccines that we make are really not optimized for intranasal delivery. So we do not use them. But I do know that there are some laboratories that try to generate formulations that are really optimized for intranasal delivery, and I'm pretty confident that in the coming years there will be a great progress in this field as well. So again, great question. We will probably need to wait a little more, but again, my expectation is that in a year or two there will be mRNA-LNPs optimized for intranasal delivery.
So as a follow-on question, that I think you started to address this, but the next question we have is, what was the rationale for immunizing mice via the intradermal route?
Yeah, that's a great point. So the reason is that when we started to test messenger RNA lipid nanoparticle vaccines, we tried many different types of routes of administration. So we tried subcutaneous, we tried intradermal, we tried intramuscular, and then it turned out that intradermal is, at least in our hands, it seems that intradermal is the best way to go. Don't get me wrong, intramuscular and also subcutaneous immunizations also work well. But for intradermal, we had to use a little lower dose and still induce the same level of immune response compared to intramuscular. In most studies, we started to switch back actually to intramuscular because this is the most common route for vaccine administration. Again, we probably need to use a little higher dose for intramuscular to get as good responses as with the intradermal. Most probably the reason for that, and the original rationale was that we were thinking and thought that okay, the skin is is full of antigen presenting cells, Langerhans cells, different types of dendritic cells, and we thought that if we do intradermal immunization, we can reach those cells and we can induce a pretty potent immune response. And actually, this is what we see. But again, intramuscular is just is almost as good as intradermal.
All right, thank you for that. This next question, you may have covered some of this already, Norbert, in the first question, but an auditor would like to know if you could compare dosage and safety profile of egg, cell, and mRNA-based flu vaccines.
Yeah, that's also an excellent point. In terms of the dose, if I remember well, Fluzone, which is an inactivated influenza vaccine, the quadrivalent Fluzone contains 4 different inactivated virus strains, and if I remember well, it contains 15 microgram HA per component. So I think it's kind of comparable to an mRNA-based flu vaccine, because we talked about it earlier that I think the mRNA vaccine dose is somewhere between 30 to 100 micrograms, so they are, I believe, in the same range. One important note is that it's kind of hard to compare these doses because in case of the inactivated flu vaccine, we talk about protein dose, and in case of the messenger RNA, it's mRNA dose, and we do not know exactly how much protein is made from the messenger RNA. But anyway, they are roughly in the same range. In terms of the safety profile, these inactivated flu vaccines have a very good safety profile, and we've been using them for decades. For the messenger RNA, I mentioned this earlier, that the nucleoside-modified messenger RNA vaccines, messenger RNA lipid nanoparticle vaccines, have an acceptable safety profile, but I do believe that we need to optimize these vaccines, and somehow we need to reduce the reactogenicity and retain the immunogenicity of these vaccines. I believe it's going to be possible to do that.
All right. And next question we have, an audience member says, although LNP provide adjuvant function, does it cause most adverse effects in mRNA vaccines?
So the answer is most probably yes. But we need to further investigate it. So that's something I touched a minute ago. That most probably the reactogenicity, and actually it's still a question if the reactogenicity and immunogenicity of a vaccine go hand in hand. I really hope that this is not the case, because if so, then it will be complicated very hard to reduce the reactogenicity without losing the immunogenicity. Anyways, I do believe that the most probably the LNP component needs to be optimized to reduce the adverse events. However, I have to mention too that based on our studies, our published studies, and others also had similar findings, it seems that the messenger RNA component, or the degradative products of the antigen encoding RNA, is also sensed by the immune system. We do not know if it specifically causes any adverse events or not. It's kind of hard to dissect that, this comes from the RNA, this comes from the LNP. But again, my guess is that it's primarily the lipid nanoparticle.
All right, thanks for that. The next question we have, it says adjuvants like AS01 and CpG, which are currently used in many protein-based vaccines, which are relatively superior to Adavax. What are your views on these adjuvants compared to LNP as an adjuvant?
So excellent point. So I completely agree. It will be very important to compare these adjuvants to the lipid nanoparticle adjuvant. I've never seen. Actually, there are a couple of papers published by Harry Friedman's lab from Penn. He used alum plus CpG with protein vaccines and mRNA LNP vaccines. So it's not directly comparable because the good comparison would be CpG plus protein or LNP plus protein. So there is no, to my best knowledge, there are no or very few papers that looked at it. Personally, I've never looked at it, but again, this is a great suggestion, and that's something that we need to do in the future. Just one more very important point that it's not going to be easy to do that, or it's going to be probably a pretty big study because what we need to emphasize that different LNPs have different like level of adjuvant activity. So some LNPs are much better than others. So this and that's why again I can imagine that you can find LNPs that are not as good as CpG or CpG alum or even other potent adjuvants, currently used adjuvants, but I can imagine that some LNPs are better than any other currently used or unknown vaccine adjuvant. Again, these studies need to be done, but this is a great point.
So Norbert, as a follow-up to that, what components of LNP confer adjuvanticity?
So based on, excellent question. Based on our studies, we see that if we, and this is what we published in the immunity paper, that if we leave out the ionizable lipid component of the lipid nanoparticle, then we lose the adjuvant activity. So most probably the ionizable lipid component is critical for the adjuvanticity. But I can also imagine that it's possible to play around with other components and increase the adjuvant effect of LNPs. Again, this is something that we are very actively investigating at the moment.
All right, and I think we have time for one last question here. An audience member says, where do dendritic cells come into play when handling mRNA vaccines?
Another fantastic question. So at the moment, we still don't know how. So what happens after mRNA administration? So it's possible that the messenger RNA lipid nanoparticle traffics to the let's say the draining lymph nodes, and in the draining lymph nodes cells take it up and translate it, and it induces an immune response. But it's also possible that dendritic cells take up the RNA somewhere in the periphery at the site of the injection in the skin or in the muscle, and then they produce protein from the RNA, and they basically migrate to the draining lymph nodes and induce an immune response. At the moment, we really don't know how mRNA LNPs reach the draining lymph nodes. So again, maybe, or it's also possible that both scenarios happen. So dendritic cells take them up and bring them to the lymph nodes, or they just traffic to the lymph nodes without dendritic cells. So so it would be nice to put together some studies that look at it.
All right. Well, thank you, Norbert. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Gen website at genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues. I'd like to thank Norbert again for his wonderful presentation, and I'd like to thank you, the audience, for your attention and very thoughtful questions. A very special thanks to TriLink for sponsoring this webinar. Hopefully we'll see you again at another webinar in the near future. Keep an eye out for the next webinar in this series, which is coming up in a few weeks. Everyone have a great weekend. Stay safe and healthy.